Okay, hello and welcome to our first Thinking Beyond of 2024. We have several events coming up that we wanted to share with you. The first being that acclaimed philosopher and artist Jonathan Keats will be visiting, and he will be hosting a workshop, and we'll have a public conversation with him. And those are both in-person only events, and they are on February 15th. Registration is open on our website for that. And then for those who may not be local, we do have our next Thinking Beyond scheduled for February 26th. And our speaker is Andrew Jordan, and his talk is What Happens When a Quantum Measurement is Made? Some New Ideas. And then our March 25th Thinking Beyond, we will have speaker John Maddock, and his title is The Dark Matter in Our Genome. And then we are very excited to announce that our 2024 Beyond annual lecture speaker is Brian Green, and that will take place on March 14th, and that's in person only as well. And his talk is called Until the End of Time. So more information and registration links will be sent out over social media and email. But you can also find the information on our website, beyond.asu.edu. And then tonight's webinar is being recorded, and it will be uploaded to YouTube later on this week. If you have any questions during the presentation at any time, you can submit them using the Q&A feature at the bottom of your screen, and we'll get to those at the end of the hour. And so that's all I have. So I will turn it over to the moderator for the evening, Beyond Center Deputy Director, Sara Walker.
Hi, everyone. Thanks so much for joining us this evening. We have an exciting topic today talking about whether there's life on Mars and how we should go find out. I'm joined today also by the director of the Beyond Center, Paul Davies, who is a very distinguished scientist in his own right and will join in on the conversation later. But our special guest today is Dr. Jan Spacek, who's a senior scientist based in Florida at the Foundation for Applied Molecular Evolution. He's a molecular biologist, analytical chemist, and organic chemist who focuses his study on life on Mars and the clouds of Venus. He's inventor and lead designer of the Agnostic Life Finder, ALF for short, and manages the Alpha Mars group. He proposed a hypothesis about organic carbon cycle in the atmosphere of Venus, which will be tested by the first private interplanetary mission in 2025. And he's also involved in developing life detection devices, including ones that can detect coronavirus, among other things. So pretty extensive background thinking about tough questions and how to detect life. In addition to that, he has a extensive visiting experience across Europe, but I think something that's kind of nice and outside of science is that he's bicycled the Baltic Sea and the Australian continent, 75,100 kilometers in all. That's pretty impressive, actually. So I'm going to pass it over to you, Jan, and we're really thrilled to have you here tonight and learning more about your work and thinking about the possibilities of life on Mars and how we're ever going to figure it out, what the right solution is. So thank you.
Yeah, thank you, Sarah, for the introduction, and thank you all from Thinking Beyond to invite me. As Sarah said, I will today be talking about life on Mars, that it likely is there, but it's up to private organization to search it, to look for it. So one thing you need to know before I get into this talk, we have a new space race. It's a Moon to Mars. NASA organizes Artemis, but Chinese are onto the race. Bill Nelson had a talk now 2 years ago that we will put humans on Mars before year 2040. China wants to beat that timeline, getting much sooner in 2030s. And of course, SpaceX and other private agencies aim to send humans to Mars quite soon. Another thing you need to know, these missions that aim to send humans to Mars will need to use local water to make a propellant for ascent rockets to get back from the surface of Mars, and for human consumption. The easiest way how to get to the subsurface ice on Mars is to drill, then melt water using what's called a rod well, where you just pump down hot water and extract the melted Martian water. So in that way, you can even on Mars easily gather large quantities of water robotically and remotely. So these missions aim to target the mid-latitude subsurface ice, which is known to be on Mars under less than 1 meter of soil, of dust. We know about it from satellite observations, but also from we can see ejection of fresh ice from impact craters, which then sublimates over time. So we are very certain that there is a lot of subsurface ice. Recently, there has been detected subsurface ice in close to equator, quite large deposits, but that's under much thicker overlaying soil, so it's harder to get to. So the mid-latitude ice is the most likely target for the crewed missions. So now for the life on Mars, I'm going to really breeze through this part because I will focus more on the detection, but NASA community or astrobiological community throughout the world is convinced that life is likely on Mars. It's either in deep subsurface where it will be deep enough where water can still be heated, melted by the geothermal heat. It can be found in caves where you have better microclimate for the microbes to live, or in salts close to surface. What's most important, most interesting for me is the mid-latitude subsurface ice, which might contain Martian indigenous life. So on Primordial Scoop, that's our blog, Stephen Banner and I have proposed some models how the Martian life can exist. So I would recommend you to visit that. I will just advertise it here for you to look for later. But what's important part is that seeking life on Mars before humans arrive there have advantages. Some we may still not understand, because Mars is the only extraterrestrial astrobiological target that will be affected by human presence in the next decade, or in the next decades, if we are less optimistic than SpaceX. We have quite short timeline, and the problem is that biological contamination, when we get humans to Mars, might make it harder to search for the extant life on Mars. And we know that there is, or NASA and COSPAR is very cautious about the contamination back and forth, because there is a non-zero possibility of some adverse effect of Martian life on Earth or other way around. So we should be cautious. We should go to Mars boldly, but not blindly. We should know what we are going to do. So, and yet we are not looking for life on Mars. So far, instruments on Mars would miss life we expect to be there. So if instead to Mars, the rovers, Curiosity, Perseverance, and the planned European Rosalind Franklin, which was ExoMars rover, were sent to Atacama Desert instead to Mars, they would miss microbial life in the soil. So this paper describes this finding. Carol Stoker wrote this essay paper discussing that it's very important to not confuse lack of evidence as evidence of absence of life, and we really need to start looking. The last missions to Mars, which actually had a goal to find life on Mars, were Viking missions in 1976. So NASA is still yet to deploy an effective tool to search for life on Mars, which is quite surprising. I assume that public usually assumes that NASA is already looking for life on Mars, but that's not correct. Now we have sample return, and sample return will be very interesting for geological purposes. Might tell us something about ancient life on Mars, but is very unlikely to tell us anything about life which is currently living on Mars, because we are sampling one of the least hospitable places on Mars. So now it's an open secret. Life would not be detected by the sample return. And so astrobiologists know about this problem, and there is a lot of NASA and international astrobiologists urging NASA to do something about this, urging NASA, telling we need to look for life on Mars, such as here Carol Stoker and her co-authors presented basically what I summarized. We have life can be in the deep subsurface, mid-latitude ice, in the caves, or on the salt deposits, and we should look for it. Here are the co-signers. A lot of very respectable scientists agree with that position, and I'm using this webinar. I know that it's going to be recorded, so you can read through it. I'm not going to read through it, but just the first sentence. A search for evidence of extant life on Mars should be conducted within a decade. That's how Carol Stoker starts her paper. And yet, national academies needs to remind NASA that NASA is not looking for extant life on Mars, right? It's quite crazy, but that's how it is. So again, this finding, NASA has not yet developed a planetary protection plan and related research activities specifically tailored to understand and mitigate the risk of forward and backward contamination for human missions on timescales consistent with the earliest plausible human missions. So basically, we need to look for life on Mars soon. And so we have actually a mission to look for life on Mars, and this is Mars Life Explorer, in short, MLE, or as Amy Williams call it, Emily. But the planned launch date is 2040. So here is Amy. She actually works in Gainesville, so she's a fellow Floridian. So the proposed Emily mission will drill the mid-latitude ice, will detect organic materials, organic molecules, detect non-equilibrium gases, and analyze depletion of isotopic ratios. It's still instrument. The instruments are still to be selected for this mission. So it's instrument agnostic, and but the problem is that these biosignatures are ambiguous. So after this mission is conducted, it might take a couple of years before we decide that if we found life or not. So, and again, the launch date is 2040. It's at the time we already might have humans on Mars, so it's quite irrelevant from that perspective. Okay, so we have a problem. People on Mars, before we know if Mars have active biosphere. So who you're gonna call if you have a problem? If you had a ghost problem, you would call Ghostbusters, but we have alien problem, so need to find alien finders. NASA has these research coordination networks that's supposed to target each individual topic. And of course, for searching for life on Mars, you would go to this NFOLD, which is Network for Life Detection, which is supposed to search for evidence of life beyond Earth. But from my experience, NFOLD actually discourages researchers from search for extraterrestrial life on Mars from extant. Extant means that the life that is still living. And NFOLD team has different research priorities than extant life on Mars. They focus on other moons and exoplanets and other targets. So from personal experience, I know that saying that we should, basically what I'm repeating over and over, what Carl Stoker said, that within decade we should find life, search for life on Mars, they don't like to see that. So, and in the paper, which I'm going to talk about more in the next slides, they removed parts of text where I was suggesting that that should be one of the priorities. So this is back to the different priorities. There is a very little overlap between the Carl Stoker's white paper and co-signers. There's only one guy who is on both network for life detection and signed this. So the question is, if Carl Stoker did not invite anybody from Unfold team, or if the if she invited the people from Unfold and they just refused to sign this. I will breeze through this because I'm behind my time a little bit. So this is the paper which is very contested. Well, in the community that wants to look for life on Mars, right? So all in here, brief summary. I recommend all to read this, but here's the summary: that all biosignatures from in situ observations of alien locomotion to spectral analysis of exoplanet atmosphere, instrumentation, mission design, and project proposals should follow the standardized guidelines controlled by a committee. Result reporting to public should be supervised also by a committee. So, of course, many scientists did not agree with this position. Stephen Banner, which is my boss, Carol Cleland, philosopher. Now, David Greenspoon is chief astrobiologist scientist. I'm sure the title is different, but you can find it out. Mark Harrison, they all pointed out the flaws in this on our primordial school. The point is that throughout the white paper, Network for Life Detection basically says give up on hope on finding and studying Martian biosphere before 2050, because it all will take decades and there will be ambiguous detections, and basically discourages students from even trying. So now this white paper was commented or quite harshly criticized by the independent review of national academies. National academies criticized this paper in this report. And again, I personally have many criticism, but national academies are considered to be the highest scientific body in the United States. So the findings that are presented in this report should be studied by scientists. So I will just go read this highlighted part. The unintentional consequences of using the proposed protocols in this way, which means in a way how is described here, would hinder the open execution of science and add gatekeepers in the scientific process. And finding 5, the scientific process of multiple working hypotheses in the most rigorous and time-proven means is the most rigorous and time-proven means by which to assess evidence and claims related to life detection. The establishment of a new external verification entity to oversee assessment and publication of light detection claims could have unintended negative consequences for open science and for fairness. So again, I recommend pause this video when you watch it in a recording and read the other highlighted parts, or even better, find this report and read it in its entirety. So in my opinion, Unfold, the organization that is tasked to search for evidence of life, opposes this effort. I noticed this in 2021 on a meeting, and now I asked Unfold leaders if they reacted to the independent review, and they have not. So So my opinion is that going forward with through NASA was quite impossible. So instead, I selected to start a private organization that was also inspired by the first private interplanetary mission, which I'm working on as well. So compared to the NASA mission, this is small, much less costly, and the development is much faster. So we started in 2020 and it's going to fly in 2025. It will carry in its payload autofluorescence nephelometer, which will measure optical properties of the sulfuric acid clouds in Venus, measure size distribution and refractive index, but also it will detect the organic carbon cycle that I proposed in 2021. Here, it's a brown inventor of the, what is it? Is there organic carbon cycle in the clouds of Venus? So these are the papers I would recommend you to also study, but that's out of topic. By the way, here are the co-authors of the paper, students, and Stephen Benner. We just returned from Grand Canyon yesterday, so that's a. By the way, we have ties because Stephen always hikes in his bottom-up shirt, and we wanted to outdone him, so that's why. Okay, so now you understand that it is up to private organizations to find life on Mars because Unfold does not want to do it. So inspired by the Rocket Lab and dismayed by the Unfold, I started a private project to look for life on Mars before humans arrive there. You can find the webpage at alphamars.org. And our goal is to seek sparse Martian biosphere unambiguously in the next decade and within a single instrument. So that's the ALF, Agnostic Life Finder. So I assembled International Association of Astrobiologists and Volunteers. We advocate for search for life on Mars before we send humans there, basically highlighting what Carol Stoker said in her white paper. And we actually have a plan how to do it with our instrument, which is the Agnostic Life Finder ALF. Here is the team. We have many professors and students helping us, and to make it more official, more efficient, we started a non-profit organization with these students in a board of directors. They are very excited and work on this project. Another good news is that we got a grant funded by NASA, which is by NASA Innovative Advanced Concepts. So we were selected as one of the few projects to receive this funding. So the goal is to develop ALF device that will be, as I said at the beginning, before we send humans to Mars, we will be mining Martian ice or melt Martian ice to extract the water that will be used for propellant and for human consumption. And ALF is designed to sit on the pipe of the water, extracting genetic polymers from the stream. If the aliens have DNA, we will extract the DNA or RNA. If they have polycations as their genetic molecule, we will extract that as well. We'll get into details in the next slide. But the main point is that we just combine the ISRU, which will be happening regardless, with the life detection. Yes. And the NEAC proposal is to advance our instrument from tabletop instrument to field-ready instrument to be tested in Arctic. So again, reminder about the mid-latitude ice. Just like Martians, like the ice, so do the humans that will fly there. So the missions that will land over the mid-latitude ice will likely encounter the Martian life. Again, I would recommend reading this. But the point here is that the mid-latitude ice was deposited in the last 100, well, during the high obliquity period, which were the youngest high obliquity period was about 100,000 years ago. So how do we find life agnostically and conclusively? So instead of biosignatures that might be produced by life, ALF searches for biosignatures that are necessary for life. So in all kind of life needs a genetic molecule. We on Earth have DNA, of course, and genetic molecules must have these negative or positive charges. Main point is that they have to be polyelectrolytes. So if we find a polyelectrolyte molecule, polymer, which contains regular genetic building blocks, we can be quite sure that we found a genetic molecule of life as we know it, or alien life. And as we know from molecular biology or physics, polyelectrolytes migrate in electric field, which means that we can concentrate them or move them using electrophoresis, or in our case, we are using electrodialysis, which means movement of those charged molecules over a membrane. This electrodialysis is used in industry quite often, and it's used for tons of biomolecules. For example, when they remove acids from cranberry juice or separate proteins in milk to different fractions. So this technology is already established for large volumes, which will be needed for those large Martian samples. So we already have built ALF, which works in our laboratory. This one moves DNA from channel 3 to channel 2. For purposes of this talk, I will not go into detail, but the point is that here these vertical lines are membranes, which allow different molecules to pass through based on their size. So DNA is small enough to pass through this first membrane, which is microfiltration membrane, but is retained by the nanofiltration membrane. So it's concentrated here in channel 2. We had a large polycation, it would move to channel number 4. All right, and these are the electron microscope photographs of these membranes. And this is the ALF prototype in a real world picture. So from the prototype I showed you before, we need to go to a flow-through model where the central compartments are constantly, where the sample is constantly flowing through the compartments, and we can concentrate the polyelectrolytes, which means polyanions and polycations, in 2 separate streams, separating them from particulates and desalting them in the process. So here is an animation how the thing works. We expect that on Mars, as we are mining the subsurface ice, it will contain a lot of particulate matter, which are these brown blobs. They will go right through because they are not affected by the electric field between these 2 electrodes. Whereas if there is some DNA, by the way, this is sonicated stream. So if there are cells, the cells would be first destroyed, and then the components of the cells would be separated based on charge and size. And of course, if there are alien molecules with polycations, or let's say some peptides or proteins with positive charge, they will be isolated in this channel. So now these students were helping with development of this instrument in summer 22 and 23. And for the NIAC, we are looking for another student. So if you are interested in developing this instrument, just let me know, and we can have you for a summer. So we will be finding life, well, that's the plan, finding life with an unprecedented sensitivity by extracting genetic polymers from tons of Martian ice. So because of such a large volume, we are amplifying the sensitivity of any downstream analysis. If somebody has a great mass spec, we can include that. Or if you want to use nanopores to sequence the concentrated DNA, we will be preparing that sample for you. So if we concentrate from 10 cubic meters, that's a small swimming pool, to 10 milliliters, we are improving sensitivity of any analysis a million times. So that will give us very high confidence if there's any life on Mars. Yeah. And yeah, to refuel Starship, you will need hundreds of tons of Martian water, and ALF will be designed to analyze that volume. So in conclusion, Alpha Mars sees the study of the current Martian biosphere as the most important task for astrobiology, and in the new space race, no other astrobiological target will be affected by humans arriving to Mars. Finding alien life will be the biggest discovery of the 21st century, and Mars likely holds life. Hopefully, NASA headquarters will soon recognize that search for extant life on Mars should be moved up in the list of priorities. I've been told over and over that search for life on Mars is a priority, but it's at the bottom of the priorities, of the NASA priorities, so it needs to be moved up. And more than consensus and committees gatekeeping science, NASA needs fair competition of ideas and clear goals. And I believe that private missions, such as the one that we propose, can provide a competition that will improve how science is done in NASA. And hopefully soon that national academies will not need to tell NASA how science should be done. And yeah, this is 31 minutes, perfect timing.
Excellent.
I'm ready for questions.
That was fantastic. I'm also like down with gatekeepers, new ideas, great. So very exciting and very comprehensive talk. So now I'm going to invite our audience to ask questions. So for those that are new to our Thinking Beyond series, there is a Q&A that you can enter your questions in, and I'll read them out to our speakers. So please do, put any questions you had during the presentation or questions on related topics. We're happy to discuss it all. So we do have a couple questions in the chat already, so I'm going to start with those. The first one comes from DJ King, who asks, what are the factors that make life on Mars so likely?
Okay, great question. So, well, scroll back. So the factors is we basically learn a lot of new things since the first time we tried to look for life on Mars, which was in 1976 with Viking. We learned what extremophile life on Earth can survive, and conditions on Mars are not as harsh as one might think. So it's not that, it's both. So from things what we learn on Mars, and from things what we learn how life can survive on Earth. For example, we know that life in permafrost brines, which are permanently below 12 degrees of Celsius, life can live there in anaerobic conditions, basically surviving in a small pocket between ice crystals. So that was quite a big surprise. And yeah, basically the reason why we are convinced that life could be on Mars is because we keep learning more and more how resilient life on Earth is, and life can adapt to conditions which we previously thought were impossible for life to survive.
Excellent. Yeah, we have a number of questions coming in, but I also do want to give Paul opportunity to ask his questions as our co-host. So I'm going to switch over to Paul now. And for those of you entering your questions, keep entering them. We've got lots of exciting stuff coming up. Go ahead.
Thank you, Sarah. I should try to be brief. Could I first, Jan, That was a very nice presentation.
Thank you.
For the benefit of those who don't know, can you just explain what is a polyelectrolyte? That's what you'll be looking for on Mars. And
Yep.
why is that a signature of life? How does that work exactly?
Yeah, you are right. I just breezed through it to fit into half hour. So polyelectrolyte DNA is a multiply negatively charged polymer. So poly means that it's polymer. Polyelectrolyte means it's either polyanion with repeated negative charge, or it could be polycation with repeated positive charge. The reason why these polymers needs to have these charges, these repeating charges, is because if the charge is missing on the long polymer, it will collapse into globular shape, just like proteins. So when we try to make DNA analogs through synthetic biology by deleting these negative charges on these phosphates, the DNA can form double helix, which can then separate and allow DNA replication and propagation to next generations. So this, if you delete those charges, then those molecules are unstable if they are longer than about 10 or 20 base pairs. So,
right, but your equipment, well, it doesn't have to be DNA. That's the beauty. Any sort of informational molecule that doesn't fold up into a mess is going to have this feature of repeating electric charges, and you'll be able
correct. So we will capture mostly DNA and RNA in this channel, but also some smaller proteins that have prevalently negative charge and can pass through these pores will be concentrated as well.
Because this raises the big question, and we love to deal with big questions in the Beyond Center, which is it's all very well going to Mars and discovering life. Wouldn't that be great? Everyone would be very excited. But if it turns out to just be boring old Earth life, then it doesn't tell us, it doesn't answer that big question we'd all like to address, which is, are we alone in the universe? Can life happen in many places? Is there a second Genesis somewhere? And the reason I say that is because Earth and Mars trade rocks all the time at ASU. If anyone is visiting, or some of our own students are listening, do go to the meteorite exhibition in ISTP4 and have a look at the Mars meteorites. So we know rocks go back and forth, and I've long thought that life can hitch a ride on these rocks. And so Mars may well have been contaminated by Earth life, or vice versa. It may be that life on Earth began on Mars. And so that would be exciting to make that discovery, but it wouldn't be nearly so exciting as to find life there, but life not as we know it.
Mm-hmm.
Yeah, I agree with everything you said here. Yeah, we have a lively exchange between material between Earth and Mars. Actually, between Mars, going from Mars to Earth, it's more common because Mars is smaller. It's easier from the trajectory perspective, getting closer to the sun. But big ejections like the one that killed dinosaurs, big impacts can eject material which gets all the way to Mars. So I would not be surprised if on Mars we find Earth-type life. So that would mean that there is no Earth biosphere, but we have solar system biosphere. And then it's a question how far the solar system biosphere can extend if you have really big impacts during the early bombardment. So maybe the panspermia is actually responsible for seeding nearby stars as well. So yeah, that's something to think about, but we can only speculate until we send out to Mars.
I see we have lots of questions coming in, and I've got lots as well, but I'm just going to ask one more organizational question quickly. So you're going to send a probe to Venus as it were under your own steam quite soon. That is your own company. And yet for Mars, you need to team up with a big player like SpaceX. Is that right? Can't you just send your own rocket to Mars?
Yeah, the thing is that here for Venus, you need to buy the whole rocket, right? But if you are going to Mars, and if they are going to already doing the mining, you are just renting space for one shoebox to put on their pipe, which they are going to use anyway. So from that perspective, it's not going to be that hard financially.
And are you getting positive feedback from that company?
Well, I am not getting any feedback yet.
Right.
But now that we get the NIAC funded, we might get some feedback as being taken more seriously.
Yes, but I think that the main political point you're making is that NASA is a lost cause at this stage, and that you have to go down the route of private operators.
Yeah, well, Stephen is much more optimistic than I am. He believes that there is enough time to re-educate NASA and then start conducting missions. I think there is not enough time. We need to start conducting the missions, and they will notice that it can be done, and that will be the education, the lesson.
All right, I'm going to sit quietly a bit. If there's time at the end, I'd like to come back to some of the politics around Viking and did Viking already find life on Mars, which would be is a fascinating topic, but I don't want to stand in the way of our important audience. So Sarah, I'll hand back to you.
Yeah, I would love to get to the questions, but I also just have one question because it kind of was sparked by Paul's question, and now I'm like super curious to know. So I'm going to take a liberty, but then I'm definitely going to get to our audience questions. So you were saying it can detect genetic polymers with a charged backbone, but there's examples of candidate genetic polymers that don't, like neutral charged ones like PNA. So would you not be able to detect that, or do you have modifications that enable you to detect really alternative systems?
No, because of the principle how this instrument works, we would miss those polymers that are not charged. Yeah.
Steve has arguments about why he thinks they must be charged. So which I've heard him argue. So I think it's fair to just look for the charged ones. I was just curious on a technical point.
We try to make the non-charged polymers in the synthetic biology, not just we in our lab, but throughout the world. And the polymers don't behave properly if they are not polyelectrolytes.
Understand.
They just fall into balls and cannot be replicated.
It's a much longer, interesting conversation we could have, but I'm not going to hold up our wonderful audience questions and patience. So there's a couple questions. This one is just a great mentoring question. This one comes from Annika Krenzlock, who would like to know if you could talk a bit about the 3-month NIAC internship and what that entails.
Yeah, just go to our webpage, alphamars.org. You can find contact there and send us your CV, and we would be very happy to interview you. And if you are compatible and if you want to spend summer in Florida, then we will have you. And you will work in lab with this kind of instrument, changing voltage, changing flow-through rates, playing with the electrochemistry of the instrument, and isolating DNA and alien polymers from a stream of water. And we should be able to get, when the prototype is more advanced, we have partners who want to give us Arctic cores, which will melt and detect Arctic life in those. So that's the plan for. I'm not sure if you get to that point during summer, but you will at least start the project, which will get there. So maybe you will decide to stick around for later, for afterwards.
Yeah. Great. Our next question comes from Ryan Thomas, who asks, if you do find extant life on Mars, what do you plan to do with it?
Study it more, right? We need to know if there is life, and then we need to know how it's adapted. Just finding it's the first step, and then we start a new field of just like you have marine biology, you will have Martian biology, and you will just need to study all the life.
Excellent. So this question comes from Gage Owens. You mentioned that if there are cells, they would be destroyed by the sonication. What would be the fate of a molecule like a charged fatty acid, like from a membrane? Would that be measured? I guess this goes to studying Martian biology. How much data will we get on this first pass?
Yeah. Depending on how you set up your sonicator, you can disrupt molecules, but usually DNA gets shredded by the ultrasound high and low pressure shearing forces to chunks which are about like hundreds nucleobases or nucleotides long. So with fatty acids, you will destroy the membrane, disrupt the membrane with sonication, but you will not break the bonds within the fatty acid. So the acids, the fatty acids will just flow through the system, or if they are charged enough and they are likely small enough, they will be also captured in one of these streams. We need to select the nanofiltration to allow those very small molecules to pass through, because we don't want to capture very small molecules that might not be live, right?
Mm-hmm. Yes. Our next question, there's actually two from Steve Ruff, and they're both good questions, but the first one's just about any connections with the Mars Life Explorer mission, which is targeted to launch in 2035. And so, why is it not a priority for NASA if they're already planning on launching a mission? And how does that intersect with the work that you're doing?
Okay, so Emily is targeted to fly in 2035, but it's the phrase is it will be deployed after the spending of a Mars sample return mission is finished. So talking to Amy Williams, she estimates last year she estimated it's going to be in 2039, and because of the sample return looks like it's going to be delayed even more than it's planned. So I expect this going to be shifted even more, unless there is some big change, like NASA headquarters deciding to move this in front of the sample return, in which case that would be great. And Alf can also fly on this mission as well. So adding one more experiment here, right? That would be great.
Yeah. Okay, the next question's from Bella Tafias Mora. What kind of steps should entities looking to explore Mars be taking to prevent biological contamination from Earth?
Pardon, what entities? What?
Oh, so like any organizations attempting to explore life on Mars, I think it's a planetary protection question. Like, how do we prevent the biological contamination from Earth?
I think we will not be able to do so.
That's fair.
If you want to send humans to Mars, so it's clear that it's recognized by COSPAR and NASA. Well, COSPAR is a Committee on Space Research, basically a whole world. Everybody agrees that if you send humans to Mars, that there will be inevitable exchange of material back and forth. So I don't think that at this point it's we can or we should postpone humans going to Mars. I think that in 2030s, if people are ready and want to go, they should go. But before they go, we need to know if there is life, and then maybe we'll make informed decision based on searching, finding Martian ecosystem, what to do about it. Maybe we just send humans in one way. No, I just want to do informed decision before we go to Mars. Mm-hmm. And I want to go myself as well. So.
Yeah, that's always the motivating factor, right? We have a few questions from Jeffrey and Sara, a couple sent ahead and then one in the live Q&A, but I'm going to pick one of the questions asked that that was sent ahead, because we haven't really touched on this as much, but it's about data sharing and intellectual property. And how does NASA plan to facilitate data sharing between public and private entities involved in Mars missions? And what measures will be in place to protect IP rights when promoting innovation and discovery?
Right. So with NASA, most of the missions are in public domain, right? The data, when you are doing research with NASA, you must publish your results in open access publications, and you have resources to do so. So I expect that NASA will continue with this policy. And then with private organizations, it depends on the private organization, right? So if there is somebody who wants to find life on Mars and not tell anybody, then do it. Alpha Mars will, of course, share all the information we will get, because we are a transparent non-profit organization. So if we find life on Mars, we'll tell you about it. If we don't find it, we will tell you about it and suggest improvements. So that's my point of view.
Yeah.
So I expect, because of the new space race, which Bill Nelson really likes to promote. I think the competition is what gets the politics to follow the goals of getting to Moon and to Mars, right? And I think it's on both sides. United States and China wants this competition, so go for it. I like competitions.
Yeah.
It's better than a military conflict.
Right. So we also have several questions from Barbara Temple, but I'm going to pick the one that she asked live, because I think it's a really nice sort of general question about the process of doing science. So the question is actually, isn't this rather like how scientists are discouraged from considering quantum interpretation? So I think maybe part of what she's asking is like your general experiences about being discouraged from asking different questions and pushing the science. Like, how characteristic do you think that is of science generally, or do you think there's something specific about this case? Not to put words in Barbara, but she like inspired me in a lot of thinking about this question. So yeah.
No, I think scientists should be able to do any kind of research as long as they find funding for it, right? I think even a stupid question should be explored and published, just to warn people what you've done, and that you explored that path, or that road, how we call it in English. So no, I think in this case specifically, what I don't know what you have in your quantum physics. But
well, Paul will know a lot about that community, so he could perhaps comment on how people are discouraged in that field as a contrast.
But I don't think that you are making official government bodies that would prevent people from publishing results or from applying for grants unless they go through peer review, which is done by appointed peer reviewers, right? So here NFOLD wants to establish committees which are like controlling dissemination of information of the data that scientists gathered.
I'd like to make a general point, picking up on what Barbara mentioned, and she could be referring to people who work on the foundations of quantum mechanics as sort of the philosophical edge, and that's quite a lively community at the moment, although for many decades it wasn't, but it currently is. Or those people who speculate that maybe quantum mechanics applies to life, that life is fundamentally quantum mechanical, not just in the sense of chemical bonds, but there's entanglement and superposition and other things going on. And that's considered still a very fringe topic, and certainly people are discouraged. But I think there's a big difference here. There are two reasons why certain questions may not get asked. One is just the bandwagon effect, that science tends to be driven by certain respected figures, and they set the agenda. And if you work in those sorts of fields, that's fine. And then if you strike out on your own in something a bit obscure, then that's tough. That's one reason. But the other we've been talking a lot about is simply money, is funding. And I come back to Viking. Cost a very large sum of money. The experiments which were done were either consistent with life or, in the case of the label release experiment, were positive. And yet NASA said that life had not been detected on Mars and it wasn't interested in looking again, basically. But I think this is largely a function of the budget. That Viking cost a lot of money. It didn't give conclusive results. Are they going to spend huge amounts doing the same thing again? And so budgetary discouragement, I think, is also pretty important. I think searching for life on Mars very much falls under that category. This is an expensive venture, and you need to be sure of your ground before committing to it. So I've been expressing an opinion, but would you agree with that, Jan?
Yeah, I would agree with that. There's more politics in searching for life on Mars because there's more money involved, right? So you need to convince the whole community to do your experiment as opposed to other experiments. So yeah, it's hard, but I don't think that because of the new space, the private companies that want to go to Mars outside of government, I think that's a great opportunity to do space science much less costly than how NASA does it. So what I would wish for at this point is hoping that NASA will not throw sticks and stones on us if we try to do something like that. And since we were granted the NIAC project award, I think that there are some voices in NASA that agree with our position. So yeah, there's more politics in this because it costs more money. So that's the summary.
Yeah. So I think we might have time for one or two more questions from the audience. The next one comes from Shelby Bird, and the question is, when will it be too late to launch a mission to find life on Mars? What is our window?
Yeah. Here ALF is the last resort, right? It flies on a rocket that prepares for humans to go to Mars. I would say the sooner we find it, the better, because then we have more time to study it before humans arrive. But the last rocket without the humans is the last rocket to search for life on Mars before we have humans on Mars. So it depends how eager are people to fly to Mars. So I expect that in 2030s, late 2030s will be too late. So with the sampler return, I made this cartoon. Can you see the cartoon? So the sample return is currently planned on the second half of 2030s, but by that time we might have already people on Mars completely invalidating the entire mission. So here's the joke that you can save NASA a couple of billions by just having private and Chinese astronauts to collect the tubes for you, which is very absurd, but it's becoming a prophecy.
Excellent. Well, I think we're just about out of time, so I'm not going to ask another question because we'll run over, and we obviously want to thank you, Jan, for your time today, and the wonderful presentation. So thank you very much. And thank you to our audience and all the fantastic questions. I apologize we didn't have time to get to them all, but do come back again and bring more questions, and we try to get around to everybody that comes to these events to get your questions answered. So we look forward to seeing you all next month at Thinking Beyond. And just as further announcements, Jessica announced at the beginning a few of our upcoming events. Our next event in the Beyond Center is actually an on-campus event in Tempe, Arizona, with conceptual artist and experimental philosopher Jonathan Keats, who does some pretty radical art. It really blows my mind. A lot of it is astrobiology related. He'll be on campus for 2 days on February 15th and 16th. doing a workshop for anybody interested in exploring conceptual ideas in an artistic space related to science on February 15th. And I'll be in conversation with him in the evening talking about the tyranny of time and why clocks are a human invention and how we might reinvent time by looking at different kinds of clocks in the biosphere. So please join us for that. And thank you so much again, Jan. That was really lovely to hear about the work that you're doing. Thank you. Have a great night.
Bye.